I would like to introduce you Dennis Slow, who actually a brilliant scientist, medical doctor, and who can translate the scientific knowledge definitely for society benefits. As I mentioned in all of my talk today, the family background and the school where you started never can be replaced with anything. Actually, Dennis Slow was born in Hong Kong in 1964 from a music teacher mother and a psychiatrist father. And after he went to school, he wasn't a good boy at all, as far as I learned from his history. He got plenty of time C for conduct at the kindergarten level. However, after that, he put all of his energy for science. And concerning the spark, actually, he realized that he would like to be a scientist. Is a unwilling curiosity what we had. And again, his father gave him a rangefinder camera and let him set up a darkroom in their house. So in all of the today who I introduced, they already had some first impression for science at home when they was working there. So, and when he went to school, and he went to University of Hong Kong, then Stanford and Cambridge, how introduced him, how good was Cambridge, and Lowe recalls seeing a picture of Watson and Crick standing in front of King's College Chapel in a book about famous scientists, and he believed that rather this is a very nice place. After his career, in 1983, he arrived to Cambridge, and he spent 2 years completing his preclinical medical training and the BA degree. And after he moved to University of Oxford for his clinical training, and later on he completed the medical degree in 1989 as well. And after obtaining this degree, he continued his study in Oxford. I look forward to Lowe's response to Tim concerning the differences between Cambridge and Oxford. However, what he did in science is really remarkable concerning he believed, and he is not believed, but he is counted as the father of non-invasive prenatal testing. In 1986, as a 22-year-old medical student at Oxford, actually, he saw a lecture from Sir John Bell on PCR, a groundbreaking DNA amplification technique, and he was thinking why to do invasive technique when there is a possibility somehow to do non-invasively. And actually, he confirmed that fetal DNA could be detected non-invasively as well. So he met his wife, Alice, at Oxford, where she was completing her PhD in physics. And after he moved back to Hong Kong, to Chinese University of Hong Kong in 1997, and he continued his career as well. So again, movies and again, stories. He loves movies, and sometimes a really, really frightening example can change people's mind. So concerning the film's opening post-mortem scene, he immediately decided not to do forensic pathology and purchase chemical pathology rather than doing it. And after that, the other big movie, and he will show you, I think, today as well, about the Harry Potter one. So, and when he watched the movie in a 3D version, it reminded him of 2 homologous chromosomes and gave him the idea how to sequence the fetal genome. Of course, not surprisingly, because of the non-invasive fetal DNA test, he received huge amount of prizes, and actually in pursuit of science, never leave a stone unturned. And not surprisingly, one of the most important part in translational medicine, actually the Lasker Prize, he already received it in 22. And I am more than sure that the Nobel Committee will not miss the opportunity to award him also for his fantastic work in one time, the Nobel Prize. But he didn't stop at that level, and he went further, and he's the Asia's first ever AI cover featured was in the genome research, and he had plenty of art things which made him actually very much interested in and gave him plenty of creative ideas. And I had the privilege to see a very famous art picture which he again got an idea that how the fragmentation also of DNA can occur. And he's not only did his discovery, but highly importantly, as we also heard from earlier presentations as well, that he continued his research. We usually say that everything needs to be discovered twice. When you write down the new discovery, and the second, you wait sometimes somebody who understands how to use it. But he decided to be part of both discoveries part, and he a co-founder of many different companies. And in 2017, he was awarded the Leader of the Year award of their contribution to biotechnology development in Hong Kong. And Chinese University of Hong Kong has a privilege now from 2025 to have him actually the Vice Chancellor and President. And I'm 100% sure that Chinese University of Hong Kong, among really the best universities, we're still going up on the ranking part. However, of course, plenty of times people are asking more time. I wish to have more time. But definitely everybody has enough time, but we don't need more time, we need less distraction. And I was in January, and after in March also in Hong Kong, and I still record the dinner when we had together. And during the dinner, he showed me his phone and said, yeah, I mean, I have a phone, but I just rarely use it, just only when it's necessary. And when he showed me, I realized the number on his phone. And he kindly agreed to show you. He received plenty of emails, but he doesn't even care, you know, he's concentrating on the task he wants. So just a suggestion, if you want to ask something from him, don't send an email to him. Ask it today, because this is the possibility to get answer for it. So, and it's really a privilege, and he's extremely active in the university's administration, research, companies. So to have him here is really a great pleasure for us. So thank you very much for your time. Sorry that we a little bit distract you from your work, but it's really a great pleasure to have you here. So thank you very much, Dennis, and the stage is yours.
Thank you very much. So it's really a great honor and privilege to be invited to this very interesting and distinguished conference. And actually, I'm particularly privileged to actually learn from all the pioneers in the field. And actually, just one information for you about my not checking the email. So actually, one day during COVID, I was actually having a lab meeting on Zoom. And then suddenly, there was a unnamed call on my mobile. And I answered a call. And it actually turned out it was from the Alaska Foundation. And they said that they sent me an email telling me I won the Alaska Award. Why did I reply? So actually, so sometimes this sort of thing could end up in trouble. So I was very lucky. But during the phone call, actually, that guy told me that you have to keep this secret for the next 3 months while they make a cartoon. While they make a cartoon of you. So I put down my phone and went back to a lab meeting as if nothing has happened. So anyway, just for you to know. Anyway, so this is the remote control, right? Okay, so this is a picture of myself actually taken over 30 years ago when I was a medical student in Oxford. So as a medical student, like many of the students here in the audience, we have to study different subjects. And one of those subjects is obstetrics. So learn about how when a woman is pregnant, she would like to know about the health of baby, including a genetic health. But at that time, to do that, you need to stick a long needle into the uterus through a process called amniocentesis. Now this method is invasive, so every time you do that, there's a risk that you might harm or even kill the baby. So as a result, many pregnant women and their families were very worried. So as a young medical student, I was thinking, why did grown-up doctor do dangerous things like that? Is it possible I could replace all of that with a simple blood test that I can do on the mother's blood? So I was thinking, is it possible that a baby might release its cells in the bloodstream of mother? But the question is, how do you know those cells are from a baby? So I thought for a while, and I thought, well, if the baby is a boy, maybe at that juncture I can see male cells circulating in the blood. So I managed to persuade a histopathologist in Oxford to actually let me do this in the lab. And so we actually use in situ hybridization, in which we have a probe which will stain male cells. So you can see here in this blood sample the number of cells with a black dot. So those are male fetal cells in the mother's blood. But of course you can imagine if every time you have to do that, it is very tedious. And I was thinking, well, is it possible to somehow automate that process? So then I went to a lecture by this gentleman who's Sir John Bell. So he was actually, at that time, just returned from Stanford. And in Stanford, he actually learned how to do a then new method of PCR, polymerase chain reaction. And so in the lecture, he told us that he thought this method is going to change the world. So after the lecture, I came up to him and asked him to teach me this technique, so which he very graciously did. So then, armed with this new technique, so I'm like a child with a new toy, so I like to play with it. And I was able to actually use it to detect male chromosome cells in the mother's blood. And then a few months later, the Lancet actually published an editorial on it called "Is It a Boy?" Because theoretically, you can use this technology to tell the sex of a baby. And then I graduated and then worked as a junior doctor. So in those days, actually, you have to work about 120 hours a week. But in my spare time, I was thinking about this project. I thought that since I've started it, I have some responsibility to finish it. So then I decided to actually go back to Oxford and work on this full time. So this is a picture of myself as a PhD student in Oxford. So you can see that I'm sort of all wrapped up because I'm using male chromosome as a marker. But of course, every cells in my body have a male chromosome. So I'm trying to protect my samples from myself. But sometimes in life, now if you do something part-time just for fun, it's all good. But if you start to do it full-time, then suddenly you see all sorts of problems. And this is one of those issues. So when I've started working on this full-time, I realized that even though in the mother's blood there's some circulating fetal cells, but the concentration is super, super low. So after 3.5 years of work, I just cannot get a test to be reliable enough to be used clinically. So at that time, the science didn't go well, but luckily, I spent my spare time knowing my wife. So the two of us then graduated actually in the summer of 1994. And then I went back out to become a junior doctor and take a really tough examination. So at that time, that examination, they have a failure rate of 70%. So it's the first time in my life when I took an exam and expecting to fail. But fortunately, I managed to pass it. And then I worked for a few more years until 1997. So that was the year that Hong Kong went back to the sovereignty of China. So my wife and I thought there's time to go home. And so I joined the Chinese University of Hong Kong. So this is, we're in the northeastern part of Hong Kong, in front of this very beautiful Tolo Harbor. Now, interestingly, about 3 months before I were to return to Hong Kong, I saw in this issue of Nature Medicine, there were 2 papers, which says that in patients with cancer, you can find cancer DNA in the plasma and serum of those patients. And then suddenly I have a strange thought, because I thought a baby growing inside a mother is actually a little bit like a tumor growing in a patient. So maybe the baby will also release its blood, this DNA, into the cell-free part of mother's blood. So what this means is that the previous 8 years, I've been looking in the wrong place. I've been looking for cells which are super low concentration. So maybe I should be looking at the cell-free DNA in plasma. But the problem is that at that time, I've just resigned from Oxford, I don't have a grant, so I don't have a lot of resources. And I've never worked with plasma before. So the question is, how do you actually do it? So, and then I recall, a time in the UK, which I never enjoyed the food there. I mean, Tim probably would disagree, but somehow I'm brought up on rice, noodles, that sort of thing. So sometimes I actually go home and cook noodles. So what you do is that you boil some water and you put your noodles in, right? And then a few minutes later, you have your dinner. So I was thinking, how about if I take some blood plasma or serum from the mother and just boil it for 5 minutes, and then take a few drops of that stuff to be tested by PCR. And of course, you ask me, why do I boil? Because I thought there might be some nasty enzyme in the plasma and serum. If I boil it, I will kill that stuff, right? Which will then not digest away my DNA. But interestingly, when I do this very simple experiment, you can see that I was able to detect a signal in some women. It's 3, and then the following 4 are negative. And later on, when I wait for those women to deliver, I find that all the ones with signal all have baby boys, and all the ones without signal all have baby girls. So this is a first demonstration of the presence of cell-free fetal DNA in maternal plasma and serum. Now, up to that moment, when people is looking at fetal cells in mother's blood, the plasma and serum is the stuff they throw away in the sink. So that's why they have not looked at it. And then we realized, actually, this DNA is present very early on, from around the 7 weeks of pregnancy onwards. And then by the 10 weeks, the concentration reaches 15%, which is amazing because at the 10 weeks, the baby is still very small and the mother is huge in comparison. And yet the baby is working so hard to push all this DNA out. And then another thing is I was also worried that after delivery of the baby, is it possible that some fetal DNA may persist? Because there was a story that some fetal cells may persist in the mother's blood. But interesting, when I did the fetal DNA story, I've had a number of women deliver by cesarean section, so I know exactly when the baby came out. And they asked an anesthetist to take blood from me every few minutes. And you can see that the fetal DNA concentration drops super fast with half-life around 15 minutes. And this information is very important later on. For example, if you try to use this technology to monitor pregnancy-associated diseases or even cancer, it will really give you a real-time signal of what is happening to your patient at that moment. So then we thought, well, maybe I can use this technology to detect some fetal diseases. And the number one reason why pregnant women go for prenatal testing is because they're worried the baby might have chromosome disorders like Down syndrome. And of course, we know that a normal person will have 23 pairs of chromosomes, 46. And then the chromosomes, of course, are they are classified in terms of length, right? Chromosome 1 is the longest, 2 a bit shorter, etc. Now, in individual with Down syndrome or trisomy 21, instead of having just 1 pair of chromosome 21, they have 3. And conventionally, to detect Down syndrome, what you do is that you obtain a cell from the baby, and then you probe it, and then you ask, how many chromosome 21 will we have inside the cells? So in this case, you see 3 red dots, so it means that this baby have trisomy 21. And now this has a problem for us because, as I told you just now, I'm looking at DNA swimming outside cells. So the question is, how can I look at that signal outside cell to deduce what is inside a cell? It actually took us 10 years to work this out. So the answer is something like this. So imagine you have a wallet, and sometimes you have, let's say, 4 coins. And some other time, you have 3 coins. And I want to know today how much money you have in your wallet. Now, one way is to ask you to hand me your wallet, but that would be like invasive prenatal testing. Another way is to ask you to stand on a balance and see how heavy you are today. Now, theoretically, if you have more money in your pocket, you should be a little bit heavier. But that balance has to be very accurate, like measure 60 kilograms versus 60.01 kilogram. So we actually estimate that to detect Down syndrome in mother's blood with a concentration of, let's say, 10%, I need to be able to tell 100 copy of chromosome 21 versus 105, which at that juncture, if you try to do it using molecular biology technique, would be very, very difficult. Now, for example, not conventionally, if you try to detect DNA, as I mentioned, you use PCR. So PCR is an exponential amplification method. Every cycle is double. So I can tell 100 from 200, but not 100 from 105. So the question is, how do you do it? So we thought that one of the issues with PCR, if you analyze everything together, is that the mother's DNA, which is indicated as red here, is in a majority concentration. So the signal will overwhelm the fetal concentration, which is in yellow. But how about if I don't analyze it everything together, if I distribute all of this into many reaction wells, so that each well will only have one molecule or no molecule, this sort of digital analysis. And then in that situation, the baby's DNA will be on its own. And furthermore, the precision of the system is basically determined by you. You just have increased the number of molecules you analyze, and you will increase your precision. So we did this in 2007 and managed for the first time to show that I can detect Down syndrome non-invasively from a mother's blood. So we published this and very happy. But then interestingly, a few weeks after we published this paper, there's another paper from another group, you can see, with almost exactly the same title. And furthermore, the two papers were submitted on the same day, which is incredible, right? So what it means, of course, is that we have competition. But the good news is that we're probably correct, because two independent groups have come up with the same thing. So now, basically, it depends on who can push it out to the clinic first. Now, this method, even though it is promising, has some limitation. So what's the limitation? The limitation is that to do PCR, you need to design a pair of primers to amplify, let's say, a gene on chromosome 21. But the problem is that in chromosome 21, you basically have around 150 genes. So if you just focus on one, you're wasting information from the other genes. So that's not very good. So I want to invent a method which will allow us to analyze all of those sequences on chromosome 21. So at that juncture, the next generation sequencing was just coming out. But people at that point, they use next generation sequencing qualitatively to tell if a tumor have this panel mutation of this virus, have this set of mutations, etc. But they are not looking at it quantitatively. So what I wonder is, can I develop a random quantitative sequencing method? For example, you look at, there are many DNA molecules in the blood, in the top here, and then I can sequence, let's say, millions of molecules. Because I know human genome sequence, I can map each molecule to a chromosome from which it comes from. And in the bottom here, I can statistically analyze the ratios between different chromosomes and see if chromosome 21 is a little bit increased or not. Interestingly, when I did this, I find it to be very good. So I looked at 28 cases, 14 with Down syndrome and 14 normal. I can see from a mother's blood, I can see that all the cases with Down syndrome, which is in green, if the baby is a boy, or in red, if the baby is a girl, and the signal is very high compared with the normal pregnancy, which is in the other colors, the blue and orange. So 100% accuracy. So very excited, and we published this work. But interesting, actually, within a few weeks of this work, the other guy also have the paper. So I then started to be very curious. So who is the last author of this work? So Stephen Quick. So look at his website and find that he's actually from Stanford. He's a professor of bioengineering. But interestingly, he also graduated from Oxford. And surprisingly, from the same year that I graduated. So I told my wife about this, and then she went and found out this picture. And this is Steve Quick. So you can see this really incredible. And then actually, it's almost like the person sitting next to you will compete with you in 20 years from now. So, so remember his or her face. And then a few years later, I was fortunate enough to be elected to the US National Academy of Science. And then on the day the result came out, I saw my name. Of course, I was very happy. And then also on the same page, I saw his name. So the three of us decided to take a same picture in the same position. So anyway, so back to work. So now we have this prototype method, which seems to be able to detect Down syndrome non-invasively from maternal plasma very robustly. But to push this out in the clinic, we need to do a clinical trial. So I spent the next 3 years doing that. And in the end, the result was very good. You can see that basically with this technology, the accuracy is 99.7%. So we thought, well, we're ready. But the problem, of course, if you want to actually push it out to be a product, I can't do it in the university. I have to work with companies. But whenever you speak to companies about this, they ask you, do you have patents? So we have spent the last 20-odd years very diligently filing patents periodically. But unfortunately, when I first started, I wasn't very experienced. So our very first patent in this area, our university licensed to a company in the UK. And that company actually didn't realize the significance of this work, so they just sat on it for a number of years. And after that, they thought it was rubbish. And they said, I don't want it, I'll give it back to your university free of charge. So which turned out, of course, to be a very big error for the company, because this patent later on is actually a grandfather patent in that field. It was very valuable. So at that juncture, I have to start to interest other people. And of course now, if I was wiser now, I'll probably open my own company, but at that juncture, I wasn't very experienced. So I decided to actually license to another company. So in one of the conference, I met this gentleman who is Charles Cantor, who is the chief scientific officer of a company called Sequenom. So we talk and we collaborate, and eventually Sequenom licensed our technology. Now on the other side, of course, you can imagine Steve Quick being in Stanford, where professors have many commercial experience. And he licensed his technology to a company called Verinata, which was then acquired for about $600 million by Illumina. So then you can see that on one side, you have Sequenom with my IPs, and on the other side with Illumina with his IP. And whenever companies have IPs, they have fights, like weapons. So eventually there were actually 4 lawsuits. And actually fighting lawsuits is actually quite a nightmare. For example, there's a process called deposition. I don't know whether any of you have been through that sort of process. So basically, it's the lawyer on the other side can interrogate you. And every time they can interrogate you for 7 hours. And can do it up to 2 days. So imagine they have a clock here, and then you sit down and put the clock on to time it. And you said, well, you want to go to the toilet. You stop the clock, you go to the toilet, and come back and then start the clock again. So it's very, very stressful. So after the process, it's almost like a PhD vibe is like a walk in the park. So my lawyers actually told me that the position is like in football, in the penalty shootout. You are the goalkeeper. You can only lose, you cannot win. So you basically have to make sure that in your 7 hours, you do not lose any goal. And that was very, very tough. But anyway, so in the end, there were 4 lawsuits. And we won 3 of them, and they won 1. So they decided to actually settle out of court. So then we pool all our patents, and then basically anybody who wants to use this technology just subscribe to this pool and use the technology. So the technology is now called NIPT, so non-invasive prenatal testing. I know that in Hungary you also have access to this, and over 100 million tests have been done now, and it's a standard of care for many countries. Okay, so up to now, you can see that basically we work for a number of years to push this technology into practice. It actually took us 14 years to do this. And of course, you can imagine now, if you have done it, some of you might decide to relax, right? And just collect a royalty or whatever. But interesting, in my case, I actually asked 2 of my students to come to my office. And then I said, well, now we've developed this test. But I want now you to think of a completely new test, which can do the same thing, which does not use any of our patents. Start from scratch. So that would be quite interesting, right? So you do one thing on one angle, because I don't want to leave any stone unturned, so you go in another direction. So the question is, how do you do that? So we think that to do that, you need to maybe think of some characteristics which is different between the mother's DNA and the baby DNA. Now, so when you look at DNA inside cell, of course, it's a very long stretch of molecules, chromosome length. But interestingly, in the bloodstream, the DNA molecules are fragmented into millions of pieces. And so now, for many years, people thought that the fragmentation is random. But we and others have actually shown that that fragmentation is not. So now in this field is called fragmentomics, basically a study of the knowledge of the system whereby DNA is fragmented. And for example, you can look at it in different ways. You can look at, let's say, the size of DNA molecules. So let's say that you collect some plasma from mother, and you use DNA sequencing. And then you sequence and then find the length of those molecules, and this is what happens. So the x-axis is the size of molecules from 50 to 200 base pair. They're small fragments. And the y-axis is the frequency of those molecules. And you can see the field DNA is in blue, and the mother's DNA is in red. So the blue DNA is on the left-hand side of the red DNA. That means the field DNA is a little bit shorter. And then you also see a series of very interesting peaks. For example, this is a major peak of 166 base pair. Of course, many of you would then guess, well, it is nucleosome size, basically. So circulating DNA is basically in circulating nucleosomes. But anyway, the take-home message from that is that fetal DNA is short and the mother DNA is long. Now, so if that's the case, then for a particular sample, the more fetal DNA you have, the shorter should be its distribution. So that means that by using a size ratio, which is plotted on the y-axis here, we should be able to predict the fetal DNA concentration in a particular sample. And the x-axis here is to measure using genetic method, like if the baby is a boy, I can use a Y chromosome. So you can see that the two basically have a very strong correlation. And this is important because the fetal DNA concentration is the #1 determinant of the accuracy of non-invasive prenatal testing. But conventionally, if you use a genetic marker like Y chromosome, you can only do it if the baby is a boy. But now with the size, I can do it irrespective of the sex of the baby. And now it's even better than that. The reason is because, let's say if a baby has Down syndrome, the baby will have an extra copy of chromosome 21, which is pushing the short DNA out into the blood, compared with the other chromosomes. So it means if you look at the size distribution of chromosome 21, if the baby has Down syndrome, it will be looking different than if the baby is normal, or if the baby have other chromosome aberrations. So this we call size-based diagnostics. So now basically you have two methods to actually solve this. One is actually a counting method, and one is a sizing method. And of course my student was very happy because they also filed a patent, which was eventually also licensed at a very good value. So good value that one of those persons, when they graduate, they can buy an apartment in China. So anyway, so that's the fragmentomics based on size. But in addition, you can also look at the end of the fragment to see what is the sequence. So we call that end motifs. Because sometimes when an enzyme cuts, they have special preference for certain sequences. And then you can also see if the end is double-stranded DNA or single-stranded DNA. I call it jagged ends. Now, for example, let's take a look at the end motifs. So basically what you do is that you sequence DNA in plasma, and you look at one of the ends. So let's say that you look at 4 nucleotides, and each of the base, of course, can have 4 possibilities, A, C, T, and G. If you look at 4 of them, the possibility is 4 to the power 4, which is 256. That means by looking at the end motif, you can separate the DNA molecules in blood into 256 times. And then you say, well, why do I want to do that? You can do that because that will allow you to explore this new field of fragmentomics. Now, for example, one thing you might ask is, why do we have fragmentomics in the first place? Now, we think it's likely because it is mediated by enzymes like nuclease. Now, if that's the case, one way to study this is to knock out those nuclease genes in animals like mice, and then see what happens to the cell-free DNA profile. So we have basically, for example, wild-type mice, and also a series of different knockout mice in which different nucleases are knocked out, and then we sequence the blood plasma and look at the end motifs and see what happens. But after you got the sequencing data, of course, you need to have some ways to analyze it. So one way that we want to do is to use something called non-negative matrix factorization, so NMF, which is basically a mathematical approach to allow you to analyze complex system and to try to separate it into the components. This sort of method is used in fields like audio engineering. It's almost like a prism. When you have some color light, you pass through it, and the prism can separate it into components color. So basically, NMF allows us to fractionate, analyze different components of the blood plasma DNA. Now, when you do that, this is what happens. So you take some blood plasma from wild-type mice, you go through into NMF, and the NMF here will tell us that there are 6 major components. So on the right-hand side, you can say profile 1, 2, 3, 4, 5, 6, and plot those in different colors. And for example, you can see that this red color seems to be very dominant. So next question is, how do you make significance of all these different profiles. The way to do that is to look at knockout mice in which different nucleases are knocked out. Like this nuclease is called DNase 1-like-3. Some people call this DNase gamma. So basically it's a nuclease which digests chromatin. And this is DNase 1, which is another DNase which you might use in the lab quite frequently. And this thing called DFFB, which is DNA fragmentation factor beta, which is also related to apoptosis. But interesting, you can see that when I knock out this DNase 1-like-3, then suddenly this red signal diminishes tremendously. So basically what we do from here is that I can now say, okay, profile 1 is related to this DNase 1-like-3. Now, for example, you can see that in wild-type mice, there's a lot of this red signal. And normally, in the blood, about 35% of the cell-free DNA has this signal. And now, when you knock out this nuclease, it diminishes to single-digit percentage. Now, apart from plasma, you can do the same thing in other body fluid, like urine. And this time, when you look at urine in wild-type animal, instead of this red signal, you see a lot of this yellow signal, which is profile 2. And this time, when you knock out another nuclease, DNase 1, then this yellow signal diminishes. So basically, by using this work systematically, we were able now to assign DNase 1-like-3 to profile 1, DNase 1 to profile 2, and DFFB to profile 3. But up to now, I still don't know what does 4, 5, and 6 means. But sometimes in medicine, even though I do not know what it means, doesn't mean that I cannot use it diagnostically. Now, for example, we decide to actually use it to see how good they are as a biomarker for liver cancer. And this is a receiver operating characteristic curve. Okay, I plot the sensitivity versus specificity. If you have a test which is complete rubbish, the line will be plotted over here, horizontally, I mean diagonally. But on the other hand, if your test is perfect, you go all the way up to this top left-hand corner. I can measure area under curve. So basically, a rubbish test, the area under curve is 0.5. A perfect test, the area under curve is 1.0. So you can see, for example, when I look at profile 1, it gives us a performance of 0.89. Profile 2, 3, 4, 5, 6. And actually, 6 is surprisingly good. It actually gives you an area under curve of 0.97. For example, you can see here is normal individual, people who are chronic hepatitis B carrier, and people who have liver cancer. There's a gradual increase in this profile 6 in the plasma. But this is liver cancer, so we wonder, what about other cancer? So we decided to look at people with colorectal cancer, and once again, you can see there's a gradual increase from controls with colorectal cancer patients without liver metastasis, and those with liver metastasis, there's a gradual increase in this profile 6. So it seems to be a very versatile cancer marker, so we decided to take a more closer look. So this is the plot of profile 6 in normal individuals. So one thing interesting is that when you look at the different colors, it looks like to be very even, because you can see from the other profile I showed you before, usually some of the profiles, some of the color, will be more dominant. Now, if you like to play bridge, this is like a no trump type of scenario. There's no trump card, right? So why is that? So why is it you have a scenario in this one profile in which there doesn't seem to be any preference of any of those sequences? Because from our experience so far, if you're looking at an enzyme, usually they have some sort of a preference in sequence. So I'm wondering, is it possible that what we're looking at is something which is the major component is a biophysical phenomenon rather enzymatic? And we wonder what sort of biophysical phenomenon can cleave DNA, can break DNA, wondering like radiation or maybe free radicals. The question is that you need to find people who are exposed to this type of insult for you to see what type of individual will have that. And the answer is maybe people will undergo chemoradiotherapy. So for example, you can see that before chemoradiotherapy, about 6% of the cell-free DNA has profile 6. But after this, it's increased to 10. And I think this will be very interesting in the future because there are people who are genetically predisposed to have more oxidative stress. like people with glucose 6-phosphate dehydrogenase deficiency. And then you can also have also new drugs, and then maybe environmental toxins. So those will be a very interesting thing to look at. Now, so up to now, I've shown you that cell-free DNA can be used diagnostically, like in prenatal context or in a cancer context. And then those guys are short molecules. The next question is it the whole picture? Now, the reason why I ask that is this. Because up to now, most people in the world, when they look at cell-free DNA, they like to use sequencing. But when you use sequencing, most people will use short-read sequencing, like that made by Illumina. Now, if you use a short-read sequencer to sequence things, of course, what you get is short sequence. How about would that be some long sequence that we're missing? What if you look at some of the long DNA sequencer, like that made by Pacific Bioscience or an Oxford Nanopore? What would happen? So we decided to actually look into the pregnancy context. Now, if you use Illumina sequencing, this is what happens. So the x-axis is the size of molecules. The y-axis are the frequency of those molecules. And you can see Illumina sequencing basically shows us most of DNA molecules is less than 200 base pair, with very few above that. But strangely, when you use Pacific Bioscience sequencing, suddenly you see a whole population of super long cell-free DNA, which are kilobase in length. And those molecules basically have been ignored in the field for the last 25 years. And actually, they're not rare either. For example, in pregnancy, in the first trimester, you can see that the median is about 15%, rising to 20% in the second trimester to about 30% in the third trimester. And then you say, what is the significance of having those super long guys? Now, one reason is maybe you want to elucidate the genome of a baby. Now, because remember, in our genome, about every 1,000 base pair, there is a single nucleotide polymorphism. So in the old days, if you look at molecules, which is 160 base pair, you need 7 or 8 of those molecules before you might hit a single nucleotide polymorphism. But now, with super long cell-free DNA like this one, it's 16 kilobase. One molecule is enough for you to have a whole haplotype, which is a connection between different polymorphisms. And this another one is 24 kilobase. But that is in pregnancy. And you ask, well, what happened in cancer? The answer is the same. When you look at cancer, when you use specific biosequencing, you also see those super long cell-free DNA. And furthermore, in cancer, those long molecules also have cancer mutations. And then you say, what is the significance? So the significance is like, for the last 25 years in the field, by looking at short molecules, we're basically looking at short text messages, like WhatsApp messages from a baby and from cancer. Each message is only maybe 10 words, etc. But now, with a super long cell-free DNA, we're looking at a whole Word document. So imagine the diagnostic in this area is going to be very interesting. Like, for example, just now when David talked about the problem of the, let's say, the specificity in the field, I think we have super long cell-free DNA, it's going to elevate to another levels. Or when you look at epigenetic signature, previously, if you have 166 base pair, maybe only one CPG site. But now, with these guys, you might have multiple CPG sites in one molecule. So next question you might ask is, where does those super long cell-free DNA come from? Now, so we're wondering about that. And then one of my lab meeting, one postdoc suddenly showed a slide like this. And then those are, of course, a chromosome karyotype. And she said, is it possible the super long cell-free DNA and the short one might be coming from different parts of the chromosome? I must say that when she first said that, I was thinking, well, is it really possible? But nonetheless, we decided to take a look. So here we plot the chromosome horizontally, and then look at the areas where there is a lot of long cell-free DNA. You can see that it's actually generally coming from the lighter color region, which is the euchromatin. But the short molecules, on the other hand, actually like to come from the heterochromatin, which is the dark areas, which is amazing. So now, of course, you say, what is the difference between euchromatin and heterochromatin? So the euchromatin are areas when the genes are switched on. And then we wonder, well, if that's the case, maybe it is related to gene expression. So we separate the euchromatin into areas in which it's low expression, medium expression, and high expression. And then quantify the amount of long DNA. You can see that the level of gene expression increases, the amount of long cell-free DNA, which is in orange, increases. But if you look at the short DNA molecules, there's no difference. And basically from this, we now can suddenly develop a new generation of liquid biopsy tests. So here, for example, in liver cancer, we're looking at genes which are particularly switched on by liver cancer. And can see based on that, I can already get a reasonable test with error in the curve of 0.9. So I think this is important because up to now, if you talk about cell-free DNA diagnostics, people will think that you're looking at the mutation profile, you're looking at the methylation profile. But if you want to look at the gene expression profile, people have to say, well, you probably look at RNA, which is unstable, or proteins, which would be a completely different field. But now, by the discovery which I told you just now, even the DNA will tell you gene expression profiles. So we have recently actually published this work. And interestingly, the editor actually invited us to propose a cover image. And I was thinking about that. And then, of course, the most important thing here is this particular figure. So I was thinking, what does this remind me of? And then I thought that it would remind me of Hong Kong, basically. And so in the end, this is the cover image. And actually, this is a building that I live in, actually. And this is an HSBC building. So finally, in summary, hope I've convinced you that cell-free DNA has created a paradigm shift in molecular diagnostics. With it, non-invasive prenatal testing is already a standard of care in many systems. And we'll talk about an emerging new field called Fragmentomics. And we'll also tell you about some new development in super long cell-free DNA diagnostics. And finally, I'd like to thank individuals from my group for generating the data which I present to you today. So thank you very much for your time.
Thank you very much. I'm a visiting scholar from China, and it's my honor to hear you and other leading experts lecture here. And about 10 years ago, I had an NIPT when I was pregnant. And I think this examination is very magic. No pain, and someone opened the blind box for me and told me that I had a very healthy baby. And now, today, I have a very lighter question. And that's, as a mom, I noticed that in your self-introduction that you said that you always got a great C when you were a child in your kindergarten, and then you changed your mind. You got A, and you becomes a very famous expert. And then I'm very curious that what kind of things is there anybody sparks your mind? Do you have special stories shares with us? Thank you very much.
Thank you. Now, so one story that Peter doesn't know from your extensive research is that the kindergarten that I went to is actually a girls' school. So it has, every class has 40 girls and just 3 boys. So I think that probably just showed a gender difference in the naughtiness at that age. So that's why they give me a C-, which in the end, I did not get into the best school in Hong Kong, but I got into one school, which was St. Joseph College, which gave us a lot of freedom for me to explore. So I think that helps with my subsequent development. Thank you.
Thank you very much for your excellent talk. What makes the fetal DNA shorter than the maternal one?
Okay, so that is a very interesting question, which up to now, there's no complete answer yet. Now, so I think that the shortness or longness, has to do with fragmentation. And the fragmentation has to do with the nucleosome, the chromatin structure. So I think one possibility is actually this is basically a reflection of the chromatin structure. And also another thing is that if you look at the graph, the mother's DNA, the major peak is 166, which is the chromatin, the nucleosome length. The baby major peak is about 146, which is about 20 base pair difference. And of course, 20 base pair actually remind me of the linker region. So I'm thinking, you know, maybe the central part, the 146 is curved around the core histone, and then you have a linker region, which is just H1. So maybe a linker region is more easily degraded, that part, and that results in the difference. And interestingly, cancer is the same thing. So cancer also is 146 versus the 166. Thank you.
So when you showed early on that the DNA is cleared from the mother's blood very quickly, do you know about what that process is? I mean, the nuclease is in the blood. I mean, is it always turning over like that, in other words?
Yes, it is always turning over like that. So I think that, now we know that because first is from the pregnancy cesarean section, and secondly, I've done similar experiment for cancer. So during operation, when your whole tumor just removed, the tumor half-life of tumor DNA is the same thing. And interesting, if you do radiotherapy, then the half-life is a bit longer. It's like 3 or 4 days. And the reason is because the radiation needs time to kill the tumor, and the tumor releases the DNA, and then it drops like this. And then, of course, the question now is probably a combination of different pathway. One pathway is the nuclease. Another pathway is the liver, is the reticuloendothelial system. It take it up, yeah. So that's why just now David, when he gave a talk, he talked about a science paper published in 2024 in which they try to inhibit the removal of cell-free DNA to try to boost up tumor DNA concentration. And that system has two components. One is to basically overwhelm the reticuloendothelial system with liposomes. And another is to actually have antibody which tack on the DNA to make it just stabilize it. Yeah, thank you.
Any other questions? If not, then before closing our today's program, we would like to thank you again, Professor Lu, for accepting our invitation and giving the opportunity to listen to these very amazing talks. Thank you again.
Thank you.